Proteins, if you know in your Greek, it is called as proteose and it is called as preeminent of the first. So it is going to be the first macromolecule, or which is identified as the most complex nitrogenous substrates containing macromolecule. It is found in all the cells of living organisms, and mostly if you see, proteins are the elements which are involved in growth of the organism. So the main constituent elements of proteins are going to be carbon, hydrogen, oxygen and nitrogen. Very important element is nitrogen. Right. Now coming to the functions of proteins, so all of you know proteins are essential for structural element of the living organism. So it is important to maintain the structural integrity and the dynamic integrity of a living organism. So for example, structural protein example is going to be collagen, and dynamic functions of proteins we are going to say the example of enzymes, hormones, immunoglobulins, etc. Now coming to the composition of proteins, all proteins are made up of amino acids. So amino acids are found to be called as building blocks of proteins. So if you see here, amino acids have a basic structure COOH and NH2 group. So the COOH and NH2 group is the important structure of protein, and you also have the side chain group which is called as R group. Now coming to the classification of amino acids, two main classification, essential and non-essential. Essential amino acids have to be produced in the diet, it has to be taken in the diet, whereas non-essential are produced in the body. And if you see the properties of amino acids, there are different physical properties which are seen for proteins: solubility, melting point, taste, optical property and ampholyte. So all of you know about solubility, they are soluble in water and insoluble in organic solvents. And if you see the taste and optical properties, taste, if you see, all of you would have eaten fried rice, right? So the most important content in your fried rice is Ajinomoto. So Ajinomoto is an important type of protein which is found to be added for taste. So addition of this Ajinomoto, which is also called as monosodium glutamate, is an important one which is found to be giving taste to your fried rice. Okay, so next one is going to be the ampholytic property. So amphoteric property is also said to be where the amino acids contain both acidic and basic groups. So they are also called as ampholytes. Now coming to the important property which is called isoelectric pH. So this pH is defined at the pH where a molecule will be identified as a Zwitter ion. So all of you know what is a Zwitter ion, right? So both the acid and the base will be present, and the Zwitter ion is found to be having no net charge. So no charge will be seen in Zwitter ions. Now coming to the chemical properties, mainly you will see the reactions due to COOH group. So the amino acids are found to form salts with bases and esters with alcohols. So the first reaction is going to be decarboxylation reaction, where amino acids undergo decarboxylation to produce amines, and the carboxyl group of dicarboxylic acids, they react with ammonia to form amide. Now coming to the color reactions of amino acids undergo many color reactions which will indicate the presence of specific groups in their structure. So for example, Biuret reaction will indicate the 2-peptide linkage, and ninhydrin will indicate alpha amino acids. Xanthoproteate reaction will tell us about the benzene ring. Millen's reaction will tell us about phenolic group. So like that you have various color reactions which will indicate the specific amino acid group. Structure of proteins. Protein structure is organized in 4 levels: primary, secondary, tertiary and quaternary. If we come to the primary structure, it involves mainly the linear arrangement of amino acids. The amino acids are all arranged in a linear manner, which will tell us about the primary structure of protein. These amino acids are held together by covalent bonds. So the covalent bonds will hold all the amino acids together in a linear manner, which forms the primary structure. Now the characteristics of peptide bond. So the peptide bond consists of 2 different types of bond: primary bond and secondary bonds. The primary bond involves the principal linkage in all the proteins, which is the covalent peptide bond, and secondary bonds, which you call it as disulfide bond, hydrogen bond, non-polar bond and ionic bond. Now coming to the determination of primary structure. Primary structure is identified by the base of the amino acids which are arranged in the linear arrangement. So three different mechanisms or processes are involved in the identification of primary structure. Determination of amino acid composition, determination of protein which are degraded into smaller fragments, and determination of amino acid sequence. So coming to the determination of amino acid composition. So the composition of amino acids in the linear arrangement of protein can be identified by treating the polypeptide with acids or alkali. So enzymes, if you add enzymes to this polypeptide, you break down the protein into smaller fragments which are called peptides. Now these small peptide fragments can be identified by a chemical reagent called dansyl chloride. So this dansyl chloride method, it will help us to identify the terminal amino acids, N-terminal amino acids, where it will bind to this dansyl chloride and it gets hydrolyzed to produce N-terminal dansyl amino acid. So the number of dansyl amino acids formed in the reaction will tell us about the number of polypeptide chains which are present in a particular protein. Now coming to the next reaction, you call it as Sanger's reaction. So in the Sanger's reaction, the Sanger reagent is used to identify the amino acids, especially the N-terminal amino acid, where it produces the DNP amino acids. So again this reaction is identified by chromatography. Now coming to the secondary structure of proteins. So secondary structure is formed by two components, alpha helix and beta sheet. So Pauling and Corey identified the alpha and beta sheets of proteins. So Ramachandran plot tells us about the secondary structure of proteins. So if you see here, this yellow and red color structures, the yellow ones are the permitted structures and the red ones are the non-permitted structure. So there is a rotation angles which have been identified, which are the psi and the pi angles. So the psi and the pi angles are found to form the important rotation angles of the proteins. Coming to the next structure called alpha helix. Alpha helix is formed by tightly coiled packed structure where each turn of the helix contains 3.6 amino acids and space between each amino acid is 0.15 nanometer. Coming to beta pleated sheets, so this is also one of the secondary structure and it is formed by intermolecular hydrogen bonds. There are two types of beta sheets, parallel beta sheets and antiparallel beta sheets. Now coming to the tertiary structure of proteins. Tertiary structure of proteins is formed by the formation of domains. So the domains will form the basic units of protein structure and two or more domains, they form a polypeptide of 200 amino acids. Coming to quaternary structure of protein. Quaternary structure of protein is referred to as the oligomers. So all the protein structures are converted to oligomers and these oligomers are in turn formed in two forms, dimer and tetramer. Dimer you call it as two molecules which will have polypeptides, where two polypeptides they form a dimer and tetramer consists of four polypeptides. The monomeric subunits, they are formed by non-covalent bonds. Now coming to the final topic, which is going to be the classification of proteins and their functions. So proteins are classified based on their functions. Structural proteins, where the example is keratin of hair and oil. Enzymes or catalytic proteins, example is hexokinase. Transport protein is going to be hemoglobin. Hormonal proteins is insulin. Contractile proteins is actin. Storage protein is oval albumin. Genetic proteins is nucleoprotein. Defense proteins are snake venoms and immunoglobulins. Receptor proteins are hormones and viruses. Other classification of proteins is based on chemical nature and stability, where proteins are classified into simple, conjugated and derived. Under simple you have albumin, globulin. Conjugated you have nucleoproteins, glycoproteins, mucoproteins, and under derived you have two subtypes, primary derived proteins, secondary derived proteins. Primary derived proteins are called as coagulated proteins, secondary derived proteins are called as proteoses and peptones. Now classification of proteins based on nutrition. Proteins are classified into three types based on the nutrition: complete proteins, partially incomplete proteins and incomplete proteins. So complete proteins, they contain all 10 essential amino acids. Partially incomplete protein, they contain or lack one or more essential amino acids, and incomplete proteins, they lack one or more essential amino acids. Coming to the biological important peptides, there are three main types of peptides which are biologically important: glutathione, which is a tripeptide, and TRH, which is called thyrotropin releasing hormone, and last one is oxytocin.